So the first thing to do in our bacteriophage experiment is something that we've seen a couple times already, which is to make a dilution series of our bacteriophage. So I have a 10 to the negative 4 concentration of a bacteriophage culture right here. So what I'm going to do is I've got a pipette set to 1 mil or 1,000 microliters. I'm going to take the mill of this, and I'm going to add it to this bottle here, which contains 9 mils of sterile water, to make a 10 to the negative 5 dilution. Pipe that up and down a couple times to mix. So there's our 10 to the negative 5 dilution. And what I'll do after that, get a new tip, take a mil of this 10 to the negative 5 dilution, and then add it to this other bottle, which has another 9 mils of water in it. That's going to make our 10 to the negative 6 dilution. So there's our dilution series for bacteriophage. So after making the dilution series, next thing you want to do is add 100 microliters of each dilution into one of these test tubes. So starting off with, I've actually got an even more concentrated version of the phage here. We've got 10 to the negative 2 concentration. So I'm going to make a 10 to the negative 2 concentration. I'm putting 100 microliters into there. Remember to switch tips as you move between dilutions. 100 microliters of the 10 to the negative 4 dilution. And 100 microliters of negative 5. And finally, 100 microliters of negative 6. So next to each of these, we're then going to add 200 microliters of a culture of E. coli, which I have right here. So change my pipette up to 200. And then I'll do this a little bit quickly. Since I'm pipetting E. coli into all of these tubes, I can just go right down the line here with the same tip. 200 microliters, 200 microliters, 200 microliters, and 200 microliters. So after that, we can mix these up and incubate them for about 15 minutes before moving on. I just want to mention the reason for incubating these at 15 minutes at room temperature is to allow for the phage, the bacteriophage, to absorb or stick to the cell surface of the E. coli. So after our 15-minute incubation at room temperature, the next thing to do is to add our mixture of phage and E. coli to some soft molten agar. And so I've got the molten agars in here in this water bath. It's important to do a couple of things with this. First, it's important to wipe off each tube on the outside so that we don't get any water contamination. And second, it's important to work quickly with your tubes of hot agar because agar does eventually solidify. So you want to make sure that you're not working with anything solid. So briefly, what you're going to do is put your phage and E. coli mixture into the agar, pipette that out, put it into the agar, and mix it around a little bit. So then what we're going to do is we're going to pour it over the top of one of our TSY plates here. So take the agar and pour it over the top. You let that solidify on the bench, and then we'll put it into the incubator. We'll do it lid side up, just to make sure, because this agar overlay sometimes can fall off, so we don't want that. So we'll incubate these lid side up. And then you're going to repeat that for all the dilutions and incubate all these at 30 degrees.